Good afternoon and welcome to today's EMSL Learn webinar, which is focused on high-resolution characterization of mineral-associated organic matter. The EMSL Learn webinar series is designed to provide information to the scientific community on resources that are available here at the Environmental Molecular Sciences Lab for research in biological, environmental, and computing sciences. Our webinar recordings are posted in the EMSL Learn section of the EMSL website, and we invite you to visit that section to see past webinars. If you want to stay informed about upcoming EMSL Learn webinars and other things, including proposal calls that are happening here at EMSL, we invite you to subscribe to our email list and also to follow EMSL on social media. We'll be sharing links in the chat so that you can follow along. Today, our presenters, chemist Odeta Qafoku and material scientist Yaobin Xu and Shuttha Shutthanandan will present resources available at EMSL for characterizing mineral-associated organic matter. The instrumentation discussed today is available to the scientific community through the Exploratory Research Call, which is currently open and accepting letters of intent through July 9th. If you have questions today for our presenters, please post them in the Q&A section so that they can answer them during the last 15 minutes of today's webinar. With that, we will start with Odeta. Thanks for joining us.
Good afternoon, everybody. I'm Odeta Qafoku, and my presentation today should be loading soon. Here we go. Okay, so I'm Odeta Qafoku. I'm a scientist here in EMSL, and today I will present chemical imaging at micro to nanoscale using one of my favorite instruments in EMSL, which is scanning electron microscope. In this presentation, I will follow three topics that are shown in this in the slide. I'll talk about surface characterization, I'll talk about chemical imaging and spatial distribution, and also characterization of interfaces in soil. So many of you are probably very familiar with scanning electron microscope, but I would take just a little bit time to describe some of the acronyms that I will be using in this presentation. And for that, I will start first with a schematic of a scanning electron microscope, which is here on the left. In a nutshell, we have a tungsten filament that generates electrons in the top. And those electrons that are generated by application of voltage and a current are pushed through this column through condenser lenses and alignments. And then they finally hit the specimen that is at the bottom of the column. So in a schematic, again, on the right, we have the electron beam is going down, it's hitting the sample. And as a result of that interaction, we have a number of electrons that are generating from the sample. They are secondary electrons, backscattered, etc. And we have a number of different type of X-rays that also are generating from the sample through this interaction. And the way that the electron microscope works is that we have certain detectors that collecting the signal. And if we collect different types of signal, it will tell us different characteristics of the sample. So in this presentation, I will be focusing on this detector, and the SEM detector that collects the secondary electrons that are generated from the sample. The EDX detector, which is short for energy dispersive X-ray spectroscopy, that collects the characteristic X-rays that are generated from the sample. I also will talk a little bit about focused ion beam. This is an application within the scanning electron microscopes, and in which we use the gallium ion instead of electrons to interface with the sample. And because gallium ions are a lot bigger than the electrons, they have a lot of energy, they can cut through the sample. So I will show some of the application today. So the three acronyms that I will use in my presentation are SEM detector, FIB, and EDX. And what kind of information do we get from these detectors? With a SEM detector, we get insight from morphology of the sample. This is a great detector to do surface analysis. You can look at the topography of the samples that have very complex surfaces, and it has a high resolution of about 2 nanometers. This is the highest resolution that we get in the SEM. The EDX detector is all about chemical imaging. It is, as I mentioned earlier on, based on characteristic X-rays. So those are specific for every element in periodic table. So we can tell a lot about what's happening in the sample by looking at that chemical imaging. We can get also semi-quantitative data from the EDX detector, and we can also create some spatial distribution maps, which I'll show later in my presentation. And the FIB application is used for many different reasons. Yaobin also is going to talk about that, but here I'm going to show how I have used it to do a cross-section of the sample. And the goal here is to investigate interfaces, and by that I mean a micro-mineral interface or liquid mineral interface. And it can give also a view of reaction boundaries and what has happened. So we can tell a lot about what is going on with respect to reaction in the sample, looking at the cross-sections. So just a summary. The SEM images are very quickly to collect. They are very easy for interpretation, and they also makes great TOC abstract graphics, which we are asked now to provide for all the journals. Okay, so I will start with 2 case studies that I will show how these detectors and applications have been used for soil samples. And in the first case study that I'll show here is related to soil mineral weathering and mineral transformation. What you see in this picture here is a picture of a calcite, calcium carbonate. This is a synthetic mineral that has been exposed to the dissolved organic matter. And so we looked at this sample after exposure to dissolved organic matter with scanning electron microscope. The calcite has this cubic structure, and the first thing that we see based on SEM detector, we see some dissolution happening on this particle, and we see also formation of some particles that, putting all the pieces together, they should be calcium and dissolved organic matter that have bound together and form these particles. So just looking at the morphology itself, we can start saying what has happened in the sample. We also can tell what's happening on samples that have been exposed to biotic inputs. And in this case, I'm showing here a volcanic glass sample that was pristine and was deployed in soil for 1 year. And just looking at the sample after retrieval, just morphologically, again, we can say we can gather a lot of information. We can first see this fungal hyphae that is attached to the grain, and we can see some processes that happen, some etching and tunneling of the grain. Again, a lot of information can be just extracted just morphologically using SEM detector. But SEM detector doesn't, excuse me, before I go there, I will also want to mention that one additional information that we get at higher resolution using SEM detector is formation of exudates and biofilm just at the interface between fungi and mineral surface. And that is important for, especially for mineral-associated organic matter. We know that those kind of attachment play a role of how organic carbon is decomposed with time. So SEM detector tells us a lot, but doesn't tell us everything. So here is an example. Again, we have a quartz mineral grain that has been deployed in the soil for about a year. And based on mineralogy, we see some form of coating. We know that the quartz, SiO2, should not have any coating. They are very clean surfaces. So to understand more about that, we now deploy the EDX detector. And immediately, as soon as we do that, we see characterization, chemical characterization of that coating. It looks that has a lot of iron in it. And very interesting also, it has a collocation of carbon with iron. So again, this tells us more about processes that iron could be moving in the quartz grain with transport, and also presence of carbon there, either as adsorbed species of co-precipitated species. Again, I will let Yaobin later on talk a little bit how we can detangle this kind of processes in much higher scale. Going back to that first image that I showed with the fungi attached to the grain, again, we learned a lot from the SEM, but when we put the EDX detector there, we see more information. We see, for example, that there is iron particles that are surround with the hyphae, and we don't know what they are at this point. One of the thoughts was that could be biomineralization of iron. But also, in addition to that, we see the carbon map, and if we had any doubt whether this was hyphae or not, you can see how strong the signal of hyphae is, and you can see also other carbon signals at other location in the grain. To address this iron precipitation or possibly inclusion, we went and did more analysis, and now we are going and using FIB that I described earlier on. So with the FIB, what that does a cross-section, a vertical cross-section of the mineral grain, and now you are seeing the vertical interface. Here we have the fungi grain, the fungi hyphae that was initially observed from the top surface, and we also have some areas here that looks like a mineral depletion, and we see also a lot more information below this interface. So when we put everything together, then we get a full picture. We see that this mineral, when you use the SEM detector, we see aggregation around the hyphae with small mineral particles. We put the EDX detector, then we see this presence of these iron-rich particles on the surface. And when we do the FIB cross-section, then we see presence of these particles of iron. Again, they are below the surface, and they are appear to be iron inclusion. It's FIB that tells us more details about that. And if we put all this together, including some work that Danny Pereira did, so I'm giving a shout to out to Danny for using atom probe tomography to help with understanding what processes are happening there. Then we understand that those processes are driven by the need of fungi hyphae for energy and nutrients. So after all this work was done, a lot of that work was done in collaboration with Rebecca Libran. She is also back to use one of the workflows that we have developed through her work here and through EMSL engagement. This is an automation SEM EDX workflow that is for soils, and Rebecca Libran and her graduate student, Cassie Collins, are using this currently through EMSL user proposal. And I also want to mention that the initial work for this started through exploratory proposal, and that's why we came back to building this workflow for this characterization. Okay, the second case study that I wanted to talk about is characterization of soil aggregates. This is some thing that we've been working in the last year. Soil aggregates, extremely important. I talked a little bit in earlier slide about initial stages of forming a soil aggregate by attachment to the fungi hypha. But in general, the soil aggregates are architectures that contain a lot of organic carbon, biota, and minerals. And a lot of unknowns are associated with aggregates. For example, the organic carbon kinetics of decomposition, especially into microaggregates, is not very well known because the organic carbon is protected on those structures. So going forward, I will, with aggregate characterization, I will start immediately with a workflow. This is being possible through the science and technology investment with EMSL. So this has been internal work that we have been doing in the last year to develop this workflow. And going from left to right, just quickly describing that, we start with a macro aggregate that we select from soil. Then we do a staining process of this macro aggregate. The staining is specifically for organic carbon. And after that, we fix the aggregate, and it looks in some form of epoxy, and that is the sample after fixation. We take this soil aggregate fixed now, and we do X-ray computing tomography analysis. This is done in collaboration with Tamas. Tamas is able to do this analysis that gives us information about connected pores, isolated pores, and solid material within the aggregates. Then we take this sample, and then we create a number of thin sections that are shown in this figure here. And then every section is characterized by EDX, and we are building these chemical maps that are shown here on the right. Those maps are very large. They are millimeter scale. And this is a new application that has been recently developed by the EDX software, where we can now apply automation protocol to create these large area maps. So some examples of these aggregates that we have characterized. The first one that I'm showing here on the left is the large aggregate that is, the entire aggregate is being imaged. And you can see the scale bars, 2.5 millimeters. So we have about 5 millimeters across, 5 centimeters across. So this is a very large map. We can also create some high resolution maps that are shown here on the right. And what you see from these two images, you can see distribution of main elements like silica in turquoise, for example, iron in green, and aluminum in orange. But I want to pay attention to this organic carbon that you can see here in everything that is in red. Those, if you look carefully, those look like cross sections of roots, fine hair roots, and they are very interesting also because they are occupying a lot of pores within aggregate. So I want to mention here that that this going from a large-scale imaging maps to higher resolution maps is a built-in function within the within EDX maps. It's not a cropping of the image. So we can go very high resolution within these maps in the area of interest, possibly micron or submicron size, but also we have about ability to go a large area maps as well. So this is kind of connecting in microscopy the micron scale to centimeter scale. This work was done in collaboration with Ashley Deer. She had a proposal here, user proposal, and brought the samples from Alaska for another research study, but was generous enough to let us use her samples to do this analysis. The second one that I wanted to show, again, similar to the first one, analysis with large area maps, is another aggregate. This is a micro aggregate. This is done through work with Devin Ripner. This is a soil that came from Eastern Washington. Again, here you can see organic carbon in red. Here is a particulate organic matter that is trapped within this aggregate, but also if you go to higher resolution, you can see some organic matter that is organic carbon is more strongly associated with the minerals. So again, where this organic carbon is with respect to pores and also what type of organic carbon is very important to make decisions of what will happen during the transport and decomposition of this organic matter. So very important to know that detail. So I would like to finalize my presentation here. I will say that SEM can be integrated with other chemical imaging and bulk analysis. We can visualize biochemical processes by using SEM. We can look at identify microscale processes that might control macroscale observations. And lastly, we can use the automation that I mentioned earlier to address microscale to pore scale heterogeneity for prediction of soil carbon. And with that, thank you for your attention. And next speaker will talk more about these techniques.
So good afternoon, everyone. I'm Yaobin Xu, a material scientist from the EMSL. So today I would like to talk about how I use cryogenic TM to investigate the soil materials. So just now, Odeta talked about how we use focused ion beam scanning electron microscope to investigate the soil materials. Here I just want to emphasize another kind of equipment we have here is the plasma focused ion beam as scanning electron microscope, which we use the plasma xeno ion source, which compared with the conventional gallium ion beam, it has much high current density, and then we can access the very large cross-sectional area up to the 1 millimeter. Besides, this equipment also has the cryogenic stage and the inert gas transfer system, so we can investigate the air-sensitive and beam-sensitive materials as well. Besides, we can also apply the 3D slicing technique to get the three-dimensional microstructure and chemical information of the materials. So using this technique, firstly, we use the plasma focused ion beam to prepare a large area cross-section, and then we can use the scanning electron microscope detector to acquire the image, or we can use another dispersion spectrum to get the elemental mapping, or use the electron backscattering detector to get the EBSD maps, which provide the crystallographic information of the materials. And later we use the plasma focused ion beam to remove a thin slicing and then get another SEM image of the EDS map. And later we can use the computer to reconstruct those image of the chemical maps or the EBSD maps together. And then we get the three-dimensional microstructure and the chemical information of the materials. So here is one example. We successfully apply this technique to the air-sensitive and beam-sensitive materials like the lithium metal or the sulfur, which is a cathode applied in the lithium-ion battery. Here is a flash, show the three-dimensional microstructure of the sulfur cathode. We use this technique. Later we can, based on those three-dimensional reconstruction, we can get this material's porosity and permeability and also the tortuosity. Besides, we also developed the in situ capability inside of the FIB SEM, which we can apply the bias or the thermal to conduct the in situ bias or the in situ heating experiment inside of the ICM. So here, another technique I want to mainly talk about is transmission electron microscope, which we can provide the microstructure and the chemical information from the micrometer scale to the nanometer scale and eventually the atomic scale. So here I just want to make a brief introduction about the transmission electron microscope. So for this technique, we use a very high energy electron beam, usually it's 200 keV or 300 keV. And we use, we have a condensed lens system, which is comprised of the electromagnetic lens. So we can converge our beam to the nanometer scale or the angstrom scale. And then we interact with our specimen. And after that, we have the different kind of the camera or the detector to collect the information from the interaction between the high energy electron beam and our specimen. So with the aberration corrected corrector, so we can increase our spatial resolution. So in this case, so we can acquire the materials microstructure, chemical, and fine structure information at the atomic scale. This is a very unique and powerful technique to do the materials characterization. So at the PNL, we have a variety of the aberration corrected TM. So each one, they have their unique properties. And also we have the dedicated cryogenic TM. So by using those kind of different kind of the aberration corrected transmission electron microscope, so we can acquire the different kind of the information, like for the probe corrected TM. So we can get the microstructure and chemical information, and also this TM they can combine with different kind of the in situ TM holder. So for the environmental TM, we can input the gas, and then we can investigate the gas-solid reaction. And also for the monochromatic TM, which has very high energy resolution, so we can do some fine structure or the electronic structure analysis. So what kind of information we can get from the TM? So here are a few examples from my previous research. So first, based on the electron diffraction information, which is similar with XRD, we can get the crystallographic information of the materials. So we can use this technique to identify what kind of phase in the materials. And also we can image the morphology of our samples, like the defects or the secondary phase. And also this technique, we can push to the atomic scale, which here are two examples show the interface or the defect we acquired in the materials at the atomic scale. And also combined with the energy dispersed spectral and the electron energy loss spectral, we can get the all element analysis inside of the TM with a very high spatial resolution. So this example, we can push our element analysis to the atomic scale. And besides, by using the electron energy loss spectral, we can get the fine structure and the electronic structure of the materials, like the bonding environment or the valence of the different kind of the elements. So besides with most advanced 4D STEM technique, we can also get the physical information of our materials, like the electric field distribution or the magnetic field distribution, which we can also push to the atomic scale. So the TM is not only just an image technique, it's also a nanoscale reactor. So combined with different kind of the in situ TM holder, we can investigate the microstructure and chemical evolution under the external stimuli like the heating, ice, mechanic, light, or the magnetic. So here are a few examples. We use those kind of the in situ TM holder to investigate the different kind of the reaction inside of the TM. This is one example. We investigate the reaction between the silicon nanowire and the lithium inside of the TM. And also we can heat up our sample and to see the thermal stability of our materials. For some kind of the air sensitive or beam sensitive sample, we can also combine with our cryogenic holder and the cryogenic transport station to investigate those kind of air and beam sensitive materials with minimal air contamination and electron beam damage. Besides, we can also create a gas cell inside of the TM to investigate the solid gas reaction, or we can create a liquid cell inside of the TM to investigate the solid liquid reaction. Besides, we can also combine with in situ environmental TM to investigate the solid gas reaction under the different stimuli, or we can use a tomography holder to investigate the three-dimensional microstructure and the chemical information at the nanometer scale. So for the TM research, a good TM sample is critical because we mainly collect the information beside of our sample. So we need our sample thin enough. So here we have the different kind of the method to prepare the TM sample from the traditional mechanical polish method, or use a focus ion beam system, or for some kind of very beam sensitive sample, we can also use ultramagnetomy method. And also for some electric conductivity samples, we can use the electrochemical method, which is very high efficient. So here I want to emphasize one technique we mainly use to prepare the TM sample is focus ion beam. So this technique, we use focus ion beam to prepare the thin laminar, and it can also connect the SEM investigation to the TM investigation, because this technique we can prepare our sample from some specific interest area. We can firstly investigate the inside of the SEM, and later we prepare the sample from this interest area, and then we transfer to the TM and zoom into the atomic scale. So here are a few examples. We use different kind of the method to prepare the sample and then image them inside of the TM. So here, why we want to use the TM to investigate our soil materials, because the TM can provide the high spatial resolution. So inside of the soil, it's a very complicated system, including the different kind of the root leaves and magma's or the minerals. The interaction or the transformation or the reaction between them is critical for our Earth's clean and health. So those kind of the transformation and the interaction or the reaction mainly happen along the interface between them. So only the high spatial resolution we can visualize those kind of the interface and investigate those transformation and interactions. So here is a schematic diagram to show the importance of the high spatial resolution. So when at the low spatial resolution, we cannot visualize the interface. So we only know, so inside of this soil, they have the organic matters or the minerals, but we don't know how the interface looks like. But when we increase the interface, then we can visualize the interface between the organic matters or the minerals and organic matters, but still we cannot see the fine structure. And later when we continue increase the spatial resolution, and then we can see the fine structure along the interface. This will help us to understand how those interaction or the transformation or the reaction happens between the different kind of the minerals, microbes inside of the soils. So here is one example. We use the plasma focus ion beam SEM to investigate the soil materials. So just now the Odeta talked about how we use FIB SEM to investigate the soil samples, and here I just want to make a complementary to show another technique we can use to investigate the soil sample is the 3D slicing. So by using this technique, we can get the three-dimensional morphology of our soil sample. This is a raw image we acquired from the plasma FIB SEM. And besides, at the same time, we can also acquire the EDS mapping at the three-dimensional. And later we can use the computer to reconstruct the morphology of our soil samples, and also the chemical distribution of the different elements inside of the soil sample. So here I just show two elements. One is silicon, mainly from the minerals, and another is carbon, mainly from the organic matters. So later we can also use the cryogenic TM to investigate the microstructure and the chemical and the bonding information inside of the soil sample, so which we can investigate across from the micrometer scale to the nanometer scale. So here is one example. So we can see the morphology of the minerals and the organic matters inside of the TM, and also we can use electron diffraction to identify the phase of the minerals, and later we can use the EDS and EOS to investigate the chemical distribution of the soil materials. Besides, we can also use the EOS to investigate the bonding environment or the electronic structure of the different element or the phase inside of the materials. So here is just one example, show the difference of the iron oxide from the surface to the inner region. So last, I would like to introduce another technique we can use inside of the TM is three-dimensional cryogenic tomography. So here is one example. Sorry about. One example, we use the 3D tomography technique to reconstruct the mineral associated organic matter inside of the TM. So we can see this cold shell structure at the nanometer scale, and the shell is mainly organic matter, and the core is mainly the minerals. They both show the porous structure. So last, I would finalize my presentation. So as I talked about, the electron microscope is a very powerful and unique technique. Since the invention of this technique in the last 1930s, so they have lots of the applications in the physics, chemics, and biological field. And today we have the most state-of-art TM instrument, which has very high spatial resolution energy resolution and temporal resolution. So hopefully we can also use this very powerful technique to investigate the soil science as well. Also, we are also very welcome other like the environmental or the biological research. Okay, that's all. Thank you.
Good afternoon. My name is Shuttha Shutthanandan. I am a material scientist at EMSL. So today I am going to talk about the helium ion microscope. So Odeta gave a nice introduction about the SEM and then what SEM can do. So as she mentioned, SEM is the workhorse for the imaging field for several years, almost more than 70, 80 years. But it has some limitation when you go to the higher resolution. Like, if you want to image anything below 10 nanometer. And it has a very small sampling depth of field. And if imaging some low Z material, it can cause some damage also. And if you have an insulating material, you need to have some coatings. Sometimes that coatings can interact with the material and can produce some artifacts, or it can modify some fine structures. I will show you some examples later on. So to overcome this, the new scientists have developed a new technique, a new microscope. It's called helium ion microscope. So it was developed by Carl Zeiss Corporation in Boston. So it is basically the helium ion microscope is exactly similar to SEM, but instead of using the electron beam, we are going to use the helium beams. So helium ions, basically. So I am not going to go into the detail why we are using the helium ion, but basically what we are doing is we are generating the helium ions using a tip made out of tungsten. It has a pyramid structure. And then doing some reshaping and then doing some processing. At the end of the pyramid, we can get a 3-atom trimer. So when you pass the helium gas, then the gas will get ionized through this atomic field. And then we can produce ion beam from these 3 atoms. So basically, my ion beam is size of this atomic diameter. So the diameter of the helium ion beam is around 0.25 nanometer. So that's the reason we get a better, higher resolution. So what we will do once we when we created the trimer, we use one of these atoms and align through all the optics similar to what Odeta showed you. And then those beams will hit the sample. So when the beam hits the sample, it has the same process. As I mentioned, the bottom part is exactly like SEM. So instead of electron, we are hitting with the helium ions. When the ions hit that one, it is going to generate the secondary electrons. That secondary electrons is going to give you the topographic image in very high resolution. Since these are ions, it can also backscatter. So if you put the backscattering detector, we can get some Z contrast imaging also. We will lose some resolution, but still we get some chemical information there. And then if the sample is thin enough, we can use as a transmission microscope like Yaobin mentioned earlier. So what is the main advantage of using the helium microscope? So there are 6 main advantages of helium using the helium microscope compared to SEM, and I will go one by one and give some example along the way. But all these main properties can be explained simply by a simple physics, which is the interaction volume between the probing beam and the material. So here is the computer simulation I am showing on silicon sample that 30 kilovolts gallium beam, 1 kilowatt electron beam, because we choose 1 kilowatt, that's what they use for the high resolution mode. and then 30 keV helium beam for the helium and microscope. So if you can clearly see these pictures, the interaction volume between all these three things are different. The helium microscope has a very small interaction volume, then the electron SEM has a little more, and then the gallium beam has even very large interaction volume. That's the reason we don't use gallium beams for the imaging purposes. We use only for the cutting purposes. So this interaction volume is the one is going to give us two important properties for helium microscope. If you look at the SEM, when the electron beam hits, wherever the beam hits, it is going to generate secondary electrons, we call SE1. Since the electron beam, the incident beam has more energy, it can penetrate inside the sample and can backscatter or do some scattering. And it can also come to the surface and eject some other electron, secondary electron, we call SE2. So since it is coming at a little further distance, that's the reason that resolution is, resolution is got worse little bit in the higher resolution mode. So because it has both SE1 and SE2. On the other hand, if you look at the helium microscope, because the interaction volume is so small, it can only produce where the beam is hit, the distribution of the energy of the secondary electron will looks like a very sharp function, so we get a better resolution. And the second thing, it has a large depth of field because of the nice focusing, because the helium ion has a very good focusing compared to the electron beams. So here is an example of the Schwannella bacteria on silica, the same sample measured using SEM and then helium microscope. You can see the helium microscope image is a little bit crispier, and then you can see a little bit deeper into the sample too. That is giving you the higher depth of field. It's another example. This is gold deposited on carbon, and this is a SEM image, and then this one is a helium image, and then you can clearly see the helium microscope image. We can see more detail in the deeper into the sample compared to the SEM. So the second property of the helium microscope is the high surface sensitivity. So the interaction between the probe beam and the material is different for the helium microscope and the SEM, because SEM you use the electron beam, helium microscope you use the helium ion. So there's the ion solid interaction versus electron solid interaction. So the secondary electron emitting process is different between those two incident beams. So if the one incident electron beam coming from the SEM emit one AC, one secondary electron, in helium microscope, one helium ion can generate 5 to 10 more secondary electrons. So that's the reason we get more surface sensitivity and more surface features. So here is the example of that one. This is the SEM image collected on the same gold on carbon sample. And then here is the same sample collected from the helium microscope. So you can clearly see there are more surface features visible because of the high surface sensitivity. The third one, that is the very important property that really distinguishes between the helium microscope and the SEM. So the helium microscope, we can image sample without any coating. So how can that be possible? Because, again, I will quickly explain what the process is. Again, here is the same image that I showed you before. So when the secondary electrons are emitted, whether you are using the electron beam or the helium beam, both are going to emit secondary electrons. During the process, they are going to leave some positive charges built up at the surface. So if you have a conducting sample, no problem, because electrons can come from the conducting samples and then charge neutralize it. But if you have insulating samples like organics, then we have a problem, right? So this positive charge is going to screw up some of the image resolution. So the helium microscope also have a positive charge, but we are lucky because we are bringing positive ions. So what we can do, when we are bringing the positive ions, at the same time, we can put an external electron gun and shoot the electron beam to the surface of the sample. So when we have a positive charge build up, then we bring the electron beam and charge neutralize the positive charge. And doing that, alternatively, we can image the samples as it is. So we don't need to put any conductive coatings. So that is the main advantage of the helium microscope, because some people, they don't want to do anything, because I got some sample from asteroid sample. They don't want to do anything. They want to do it as it is. So we use the helium microscope. So here is one quick example of the electron beam charge neutralization in the helium microscope. So this is the protein mineral interaction with single cell organism called Peporominifera. So the scientists from University of Washington are studying this skeleton. This is basically calcium carbonate skeleton of this single cell organism. So we can find this organism deep into the ocean bed, and these are like millions of years old. So it contains a lot of climate information. So they want to study using this organism to model the climate model from the older years. So they want to study this spine-like structure on this particular organism and see how these structures are forming and things like that. So when we put the sample into the helium microscope without the E-beam, it looks really dark. Then when we put the electron beam, then you can clearly see it is bright. And now we can go in and then we can image it in a high resolution mode, and then we can study detailed analysis. So you can clearly see the tip of some of these structures. It has some nice faceted feature. So you can get a lot of information doing like this. So the second one example is I'm doing the helium microscope on the same organism with a coated with a gold and then uncoated sample. So you can clearly see the uncoated sample lost all these fine features sitting in the top of this one, right? And the second thing you can clearly see, this one, it has a more smooth surface, the coated one, compared to the uncoated sample. And then you can see there are some other features missing in this, in the coated sample too. So we believe that, if you want to do some measurement without modifying anything, then we can do that with a helium ion microscope. Another advantage of the helium microscope is to image very beam sensitive sample, like a monolayer of polymer film or monolayer of graphene and things like that. Because again, the interaction is the same physics that I mentioned. The maximum damage in the helium, the electron beam interaction happening at the surface, while in the helium beam, it happens deep into the sample. So at the surface, the damage is very minimal. So here is some example of the polymer film patterned on gold surface using the E-beam lithography. When we image with the SEM, we couldn't see anything, but when we image with a helium microscope, you can clearly see the structure. So these are just a monolayer thick polymer film. Okay. So there are a lot of examples, I can show you using helium microscope in BER field, but I'm going to concentrate only on few of them. One is the polymer mineral reaction interaction studies. In this one, the scientists have been developing some biomimetic structure that can support some bone mineral uptake. So what they did, they took some biodegradable polymers and they immersed them into this electrolyte solution mimicking the fluid of the body fluid, mixing with some ionic species. And then they want to study how these things are, how these minerals are nucleating on these polymers. So here is the image of the helium microscope, image of the mineral formation at 1 hour. So you can see there are small minerals are forming, and then initially they are forming like a semi-hemispherical, and then after the longer hours of mineralization, they are forming like a spherical particle. So if they did the same experiment on the SEM, we couldn't see the fine small structures that we can see it from the helium microscope. Similarly, there is another group, James Bryant's group. They are investigating the mineralization of the twisted stalks produced by micro-aerotic iron 2 oxidizing bacteria. Again, they want to study how these stalks are forming and what kind of structure it is forming as a function of time. So again, I'm comparing the SEM versus helium microscope, and you can see helium microscope gave you the better resolution compared to the SEM. And then we can study as a function of the time, 24 hours, 96 hours, and 4 weeks. You can clearly see the structures are growing, and then more petals, leaves-like structures are growing on these stalks. And then again, you can see similar structure on SEM, but helium microscope give you the better resolution. And here is the high resolution image of the same one of these particles here, one of the features there. So the second and maybe the last example, because of the time limitation, the aerosol. So helium microscope also being used at aerosol analysis. So this is the organic, inorganic aerosol particles collected in somewhere in India by Swarup China from EMSL. So you can see this is we collected without putting any E-beam. Sometimes it is good to have that because you can clearly identify the conductive sample versus non-conductive sample. So here is a conductive material sitting there. And then here you have some bacteria properly sitting there. And then there are some small particles sitting here, which have a conductive and non-conductive thing. So this is very useful that you can clearly identify all these small particles. Another interesting thing is the same group has collected some particles in Great Plains site in Lemont, Oklahoma. So they were collecting some aerosol particles, but while they were collecting, they found out there are some spherical particles intact sitting in that collected membranes. So they think that these are airborne soil organic particles. So these are very high in aspect ratio. And in addition to these spherical particles, they also show some of the small residues happening closer to that particle. So those residues are not well defined in the SEM images. So we decided to do the helium microscope image. And then this is SEM image collected at 75 degrees. And this is the SEM helium microscope image collected at the normal incident. Then you can see these black dots are these spherical balls here, because the contrast is different between SEM and the helium microscope when it is charging. And then you can see there are some residues formed along, some plain residue, or there are some different shade of residue, and then there is some particle without residue. So these are, they believe this is from some sulfur residues, and these are some sulfur residues coated on these particles, and the other one we don't have anything, So using the helium microscope, we were able to identify all these things very nicely. In addition, if you look at the images of these residues, some of them are darker and then some of them are smaller. That because of the thickness of this residue. So the higher the grayscale image intensity is the larger the thickness of that residue. So the helium microscope is very powerful to measure all these things. So similarly, we can see if you collect the aerosol particle, this is some soot particle collected at different depth. And then you can see, you can get some distribution of these particles. And then also you can characterize the shape of this particle. Some of them are spherical, and then some of them are different shapes. And these are very small particles. You can see these are 4 nanometer and 1 nanometer. So if you are going to do TEM on this sample, it will take longer time. But this one, you just put it in, and then you can image the sample. So since the time is the limitation, I'm going to skip most of the stuff. And basically what I am going to show is the take-home message from the helium microscope perspective. It can give you the high resolution, high depth of field, high contrast images on uncoated biological and environmental sample. So this is the important one. So we can image it as it is. So most of the time we can preserve some fine structures that we will lose some time using the coating technique. Thank you very much.
Thank you, Shuttha. I'm going to have our speakers come back on camera. And we know that we're kind of running out of time here, but we're going to ask each of our speakers one question, and then each of the remaining questions will be sent to our speakers and they will email you the answers. So nothing will go unanswered today. Okay, first question is for Odeta from Kate Heckman. Are there difficulties with getting the epoxy to penetrate the small pores?
That's a great question. I haven't seen any pores unfilled, let's say that. And it just depends also what size of pores. I mean, if we're talking micron size, I think they're getting filled. If we're talking nanometer size, I think that needs probably a deeper investigation if every single pore is filled or not.
Okay, thank you. Okay, next question is for Yaobin from Jeremy Sabo. Does anyone have experience using biological samples for any of the instruments discussed today? I'm interested in cell structure and metal localization within biological samples.
Okay. So for the transmission electron microscope, I have a little bit experience in the past to investigate the cell structure. So for those kind of sample, we mainly use cryogenic ultramagnetometry to prepare the TM sample, and then use the cryogenic transfer station to transfer to the TM and image them at the low temperature, like negative 170 degrees. Yeah. So I have a few experience, but I think at EMSL we have experienced the expert James Evans. He is an expert in this field, and I think from the EMSL website we also have a record video previously. He talked about this field, yeah, by using the cryogenic TM. Yeah, thank you.
Great, thank you, Yaobin. Shuttha, last question comes from George Herbert. The resolution advantage of the BE beam is fantastic. That was his comment. An electron beam brings the advantage of EDX elemental identification. Is there any similar capability when using a helium beam?
Yes, so we can use the time map flight or the like a nanosims, right? So we can get some secondary ions during that process. We cannot generate X-rays. That's very, I mean, ion beams can give X-rays, but since the energy of the helium beam is small, 30 kV, they cannot produce X-rays. But we can do secondary ion imaging. But unfortunately, EMSL doesn't have that capability, but some other labs has some capability. So you can get nanometer resolution of molecular information, not the elemental information like what the EDX can give. Here you can get actually the molecular information in a very nanoscale, nanoscale. But unfortunately, EMSL doesn't have that capability.
Okay, thank you.
Good question.
Shuttha. Thanks. So just a reminder as we close up the webinar that this recording will be available in about a week in the EMSL Learn section of our website. So if you want to go back and rewatch it or share it, it will be available there. Also a reminder that the exploratory call, which features the instrumentation that was mentioned today and a ton of other highlighted capabilities, that call is open until July 9th for letters of intent. So we invite you to check that out, follow up with our speakers here if you have additional questions. And for those of you who didn't get your question answered, you will hear from our speakers. We will make sure that they receive your question. We'll reach out. So thank you for attending and have a great afternoon.